I just floated in a phrase that our nurses have coined, and this was some time ago. There is no other hospital like it. I really like this phrase, and I used it as the byline for the clinical center for many years. But what makes us different? Why do we have that phrase? Well, the first thing I would argue is our vision. And our vision was actually first given to us on June 22nd, 1951, by President Harry Truman, who laid the cornerstone at the clinical center. And in that meeting, he said, modern medicine must find ways of detecting diseases in their early stages and of stopping their destructive force. That will be the major work in this clinical research center. In other words, a hospital totally dedicated to clinical research. I think that vision is one of the things that has driven us for all these years. And I point out, next year will be the 70th anniversary of the clinical center. What else makes us different? Care has always been free. We've never billed a patient for anything. We bring patients from all over the country and indeed all over the world. We're allowed to spend as much time as we need for the patients, both for their care and for understanding their disease and trying to make advances. Every patient who comes participates in one of the 1,500 clinical research protocols. These protocols are really the engine that drives the research part of the hospital. And it's a hospital literally surrounded by research laboratories, not only in Building 10, the hospital building, but in also the surrounding buildings on this campus. And one of the things I want to show you as I go through the display of accomplishments is how that interaction has really helped to nurture the wonderful accomplishments that have been made. Indeed, these labs are really special. So there have been 33 Lasker Awards and 7 Nobel laureates that have done their work at the clinical center. Although not all the Nobel laureates did their work at the clinical center, most of them did. And those that didn't interfaced in the clinical center in interesting ways. So let's now go to the accomplishments. And I would say the first accomplishment I want to share started in 1953 with the first medical board, which is shown here. And as you heard from Larry, you can see dramatically the change in phenotype of staff. Here are the clinical directors, all white Caucasian men, back in 1953. But they had some really insightful thoughts. And one of their thoughts, I think their third meeting, was to issue guiding principles in medical research involving humans that required prospective review of all human research at the clinical center, including a detailed review of the informed consent process. This policy, which was really a cornerstone for how we do research, later became the basis for all the national institutional review boards that were created by the Surgeon General about a decade later. So what else makes us different? Our special resources that enable our studies. Shown first in this list is the biomechanics lab, and you see a walking skeleton. It's actually a walking person, reduced to the skeleton and the forces of the muscles and the bones that can be studied. And I will get back to that in my last comment at the end of this talk. We have 4 metabolic chambers, which are special chambers to study the physiology of people with obesity and people with wasting syndromes. These are phenomenally valuable resources. We have a whole center of cellular engineering, which manufactures CAR T cells and other key products for some of the therapeutic strategies that are being developed. We have an MRI center attached to the hospital. This center has 20 MRI machines in it, and there are actually 10 MRI machines in the clinical center itself. And you saw there are only about 80 patients in the hospital. This is a phenomenal resource. There are 3 7 Tesla MRI machines and 1 11.7 Tesla MRI machines. For those of you into imaging, this is a phenomenal resource. Our PET research department has 3 cyclotrons for manufacturing tomorrow's probes for use in cyclotrons to look at receptors in various disease states. We have a high containment clinical studies unit where we can safely bring in patients with Ebola or COVID. We have a very strong healthy volunteer program these folks who come here serve as important controls in many of our studies. And we have a very special bioethics department where the bioethicists go on regular rounds with the physicians and care providers available to talk about and address any ethical issues related to the research and patient care. So let me now go through the selected accomplishments, and I'm going to do this by area of clinical need or clinical disease, and show you a long list of people, introduce you to a long list of people who've made these accomplishments. Let's start with cancer. So in 1958, Min Chu Lai had a patient under the care of Roy Hertz who had choriocarcinoma. And this was a woman who was going to die from her cancer. And he decided to do something new and use methotrexate to treat her. And lo and behold, she got better. She was cured. Then he did this in a few more women who had choriocarcinoma and developed the first treatment for a human solid tumor, the beginning of chemotherapy for cancer. In 1964, Emil Frey and Emil Freyreich, using chemotherapeutic approaches, cured childhood acute lymphocytic leukemia. Phenomenally special accomplishments. 1965, a series of investigators in the Cancer Institute really put chemotherapy on the map. Vince DeVita for Hodgkin's disease, Jim Holland for other childhood leukemias, John Siegler for Burkitt's lymphoma, and Gordon Zubrud, who developed the use of cisplatin as a treatment in cancer. All the people you see on this slide won Lasker Awards for developing chemotherapy for cancer. More recently, Steven Rosenberg, who has been the chief of surgery in the NCI for many years, in 1988 did his first publication on treatment of metastatic melanoma with cell transfer therapy, which was later shown to give a durable and complete response. I believe Steve is the father of cellular therapy for cancer. So this group has heard from Marston Linehan a few meetings ago, who has really pioneered the genetic basis and management of hereditary kidney cancer and helped to come up with some new treatments for these patients. Ira Paston and Robert Creekman were the first to use an immunotoxin coupled to an antibody targeting a cancer cell to treat hairy cell leukemia of patients who had relapsed and failed treatment and developed cures. Ira was a basic scientist who's migrated into clinical research. He's trained 2 Nobel laureates, Harold Varmus and Bob Lefkowitz. So it just shows how the interface of these basic science labs with clinical medicine is real. This is not just imagined. Peter Pinto, Brad Wood, and Peter Choyke were the first to use MRI ultrasound fusion-guided prostate biopsies. And this approach has now been brought to routine healthcare private practice and has changed the management and treatment of patients with prostate cancer. And James Kokendorfer and his colleagues developed a new treatment for refractory lymphoma using CAR T cells, which was recently approved by the FDA. Let's migrate now from cancer to transfusion medicine. The Coulter counter was invented at the Clinical Center by Wallace Coulter with George Breacher in 1956. This machine, if you don't know it, counts automatically the cells in the blood and is used in virtually every lab in the United States and the world. In 1961, Alan Kleiman did the first plasmapheresis and showed that this plasma was very rich in platelets. And for many years, this was the source of platelets for patients who needed them. 1965, George Judson, who was visiting from IBM in partnership with Emil Freyreich, invented the continuous flow blood separator for separating out all the components of the blood. And I want to just spend few moments talking about Harvey Alter. Harvey in 1964 was a first-year fellow, or actually 1963, was a first-year fellow at the clinical center in the blood bank, learning how to oversee transfusions. And he was interested in learning how to look for antigens in the blood. And he met Baruch Blumberg, who was another investigator at the clinical center. And together, in Harvey's, one of his very first experiments, they discovered Australia antigen. Blumberg went on to characterize that as the coding antigen on hepatitis B, for which he won the Nobel Prize. Harvey was the second author on the paper. He didn't win the Nobel Prize, but it didn't stop him. He became passionate about hepatitis, and you've heard him present. And in 1970, under Harvey's leadership, the blood bank switched to an all-volunteer donor program, reducing blood transfusion hepatitis from about 30% to 11%. And now this has become a national process. And in 1989, he co-discovered with Michael Houghton and Charles Rice hepatitis C virus, for which in 2020, Harvey won the Nobel Prize with Houghton and Rice. So again, he started with the future Nobel Prize winner, and had his whole career sort of evolve from his early studies on hepatitis, showing again the interface between the wonderful laboratories surrounding NIH and the clinician investigators. Now move from transfusion medicine to hematology, and I'll just point out in 1995, Neil Young developed a treatment for aplastic anemia with antithymocytglobulin and cyclosporin, which has now become the standard of care. Same year, Griff Rogers, who's now the director of NIDDK, showed that hydroxyurea can make a big impact on patients with sickle cell anemia, but not a cure. But in 2019 to the present, John Tisdale first initiated and then partnering more recently with Courtney Fitzhugh, they able to cure patients with sickle cell anemia with bone marrow transplant, and more recently with gene therapy. And they're now working to see if there's any way they can make this a practical, inexpensive treatment for patients in the developing world, such as Africa, where there's such a high incidence of sickle cell anemia. So we'll now move to heart disease. In 1957, Gene Braunwald and Glenn Morrow described for the first time this disease called idiopathic hypertrophic subaortic stenosis, and Glenn Morrow developed some surgical procedures to bring relief to the patients who had this problem. 3 years later, Braunwald's wife, Nita Starr Braunwald, who was a cardiac surgeon, did the first successful mitral valve replacement at the clinical center with Glenn Morrow. In 1961, Donald Fredrickson began his work with Bob Levy, describing a new disease called Tangier disease, named for patients from Tangier Island in the Chesapeake Bay, who had a terrible problem of heart attacks early in life, teenagers and 20s, and had very high lipid levels in their blood. This led them to understand the role of lipids as risk factors for coronary heart disease, which they described. And that's obviously a phenomenal accomplishment. At that time, 2 investigators at NIH, but not in their lab, Michael Brown and Joe Goldstein, working with Chris Anfinson and other labs, got interested in these patients and were fascinated by a teenage girl who died of a heart attack. And this led them to their lifelong study of cholesterol metabolism, for which they later won the Nobel Prize. And finally, I point out that in 2002, Philip Gordon described the use of leptin to treat patients who had lipodystrophy. One of the great accomplishments of the National Institute of Allergy and Infectious Diseases is the development of vaccines, along with some other institutes. In 1987, in the Child Health Institute, John Robbins and Rachel Schneerson developed the Haemophilus influenza meningitis vaccine, for which they won Lasker Awards. From 1972 to 1994, Steve Feinstein Al Kapitkian and Bob Purcell developed three critical vaccines. First, Feinstein and Purcell defined or isolated hepatitis A virus and developed a vaccine to it. Then they developed a vaccine to hepatitis B. And then working with Al Kapitkian, who was the lead, they developed the first vaccine against rotavirus, the most common cause of infant diarrhea worldwide. 2005, working on our campus, Doug Lowy and John Schiller developed the technology for the HPV vaccine, which has been critical in protecting girls from cervical cancer and men from penile cancer. And in 1997, the first Ebola vaccine was developed by Gary Nabel, working at our Vaccine Research Center in partnership with Barney Graham, who brought it to the first in human use at the clinical center. And in 2020, also at the Vaccine Research Center, Kitzmiki Corbet and Barney Graham had a critical role in developing the technology for the COVID-19 vaccine. And the first patients or people to receive it were at the clinical center, led by Barney Graham. I can't help but comment about AIDS and salute Tony Fauci and Cliff Lane, who you just heard about, for all their work defining the immunopathogenesis of HIV. They played a major role in that. Bob Gallo, working in the NCI, developed a screening test for HIV, which was used to eradicate HIV from our blood supply. The first three treatments for AIDS were developed at the clinical center. Sam Broder used AZT as the first treatment of AIDS, then 2,3-dideoxycytidine, or DDC, and 2,3-dideoxyinosine, or DDI, were developed at the clinical center. We became very interested in the opportunistic infections affecting AIDS patients. And Joe Kovacs in our critical care medicine unit did the first genetic sequencing of pneumocystis carotii and then developed rapid diagnostic tests to detect patients who had that infection. And Henry Mazur, who leads our critical care medicine department, actually wrote the national guidelines for HIV, and he's also written guidelines for Ebola and COVID later. Now, some of the other infections, Steve Strauss, who died at a very early age, sadly, did the first clinical trial of acyclovir to treat genital and oral herpes. And one of our current tenure track investigators, Samir Kadri, who's a computational epidemiologist working in critical care medicine, did a survival analysis of COVID patients in 558 US hospitals. And among the things he showed is that intra-hospital transfer of COVID patients can be safe. And most recently, Dan Chertow, another tenure track investigator, has a paper that's about to come out in the next week in Nature, analyzing autopsies of patients with COVID and showing persistent viral infections, including as long as 230 days post-viral symptoms detecting virus in the brain. I think this may have huge implications for our long haulers. Hopefully they're not infected, but it's something to worry about. A few comments about immunology. I want to salute Warren Leonard and John O'Shea for the development of the JAK kinase inhibitors to treat autoimmune, allergic, and well as the inflammatory process in COVID-19. These drugs have now been licensed and approved and are being dispensed by several companies and have completely transformed the treatment of rheumatoid arthritis, among others. And it's a very, I think, important contributions they've made. Bob Nussenblatt, working in the Eye Institute, and colleagues developed sustained release ganciclovir implants for cytomegalovirus virus retinitis. And in 1997, Julie Segre, Tara Palmer, and David Henderson demonstrated that microbial genomics can be used to understand hospital infection outbreaks. And this has really, I think, revolutionized the approach to hospital epidemiology of infectious diseases. Let me now shift to mental illness and tell you a few things. Fred Goodwin did the first clinical trials of lithium for depression and mania at the clinical center. Louis Sokoloff invented PET scanning and demonstrated that 2-deoxy-D-glucose was the principal or the most useful probe, which is still used in those scans. Judy Rappaport first described obsessive compulsive disorder in children and also developed the first treatment with clomipropine. And she also demonstrated that clozapine could be used to treat childhood schizophrenia and bring a substantial relief. And Susan Suedo described this disease called pediatric autoimmune neuropsychiatric disorders associated with streptococcal infections, or PANDAS, a very substantial contribution. But maybe the biggest contribution in my mind recently to mental health is the use of ketamine, which, as you may know, is an anesthetic, to treat depression and suicidal ideation. And you can give it to patients in an emergency room and within hours get dramatic relief. This is a very exciting observation. And recently, Carlos Zarate, who led these studies, has been using esketamine, which doesn't have addictive problems for depression. And I think these ketamine derivatives are going to be used more and more widely going forward. Peter Schmidt and David Rubinow described premenstrual dysmorphic disorder and treatment with fluoxetine, and Ellen Liebenloft described a disease called disruptive mood dysregulation disorder in children, and this differentiates irritability in children from pediatric bipolar disorder, which is phenomenally important for those children. And lastly, Marilyn Powell, the clinical director of the Mental Health Institute, developed a suicide risk toolkit used at the clinical center and now more broadly throughout the country. A few comments about endocrine diseases. Receptors on cells were described first by Jesse Roth in NIDDK, and those were insulin receptors. He also trained a Nobel laureate, Bob Lefkowitz. 1985, Lynette Neiman and Lynn Loriot described Cushing's syndrome's treatment with glucocorticoid antagonist RU486. 2017, Denny Porter described treatment of Niemann-Pick disease, a rare disease, with intrathecal 2-hydroxypropyl beta cyclodextrin. And the same year, Jack Yanofsky, who studies obesity, defined melanocortin-3 receptor and neurotrophin factor as important in obesity and potential targets for future therapy. And lastly, Debbie Merck, who was recently appointed the first chief of the pediatrics department in the clinical center, has described how to manage and treat congenital adrenal hyperplasia in children. My last slide on these topics is to just show you something about techniques and devices. First, in 1963, Ayub Omaya invented the Omaya CNS reservoir, a reservoir you put in the brain so you can deliver drugs and sample spinal fluid over a long period of time. John Dotman, the former chief of radiology, developed a method to locate parathyroid glands. Recently, like last week, Drew Manis published a paper in Science Translational Medicine describing a new low-cost 3D printed miniature ventilator that has no moving parts, no electricity, and he demonstrated its utility in pigs, and now it's being reviewed by the FDA for clinical trials. This is a device that can be used in the ambulances if it works. It can be used in the battlefield, and it could be used in developing countries. And lastly, I'll get back to the biomechanics lab I mentioned earlier. I salute Diane Damiano and Thomas Bulia. Diane is a PhD working in rehab medicine, and Tom is a tenure-track investigator who's a robotic engineer. And they were impressed with children with cerebral palsy who couldn't walk well, and developed external robotic devices that have really changed the life of these children and enable them to become integrated into society. So in summary, what I've tried to show you is that we focus on three major areas at the clinical center: studies of the pathophysiology of disease, studies using the first in human with new drugs and devices, and if we see something promising, we will often give it to industry or share it with our colleagues at the universities. And then we spend a lot of time studying rare disease. And indeed, half our protocols are about rare diseases. And you might say, why? Well, first of all, they're not so rare. About 30 million Americans have a rare disease. And we give them hope. But more importantly, I like to reflect on the fact that these patients often provide a window into understanding common diseases. And I don't have time to go into that today. That would be another talk, but it's a very exciting spin out of the studies we do.